Hello and welcome to my latest podcast. Today we're going to be talking about the plain language summary of one of my recent papers tracing the transmission of carbapenem-resistant Enterobacteriaceae at the patient ward environmental nexus. This is a plain language summary of a paper that myself and colleagues across UCL and the NHS wrote, which describes how we used sequencing to help understand how an outbreak of highly antimicrobial resistant, or AMR, bacteria were moving between patients in one of the hospitals. As a bit of a background to the study itself, the NHS doesn't routinely use sequencing as a diagnostic or a surveillance tool, but they were unsure whether these patients were transmitting the bacteria to each other or whether they might be getting it from the ward itself. And it's well known that pathogens can be moved around on healthcare workers' hands and also be picked up from surfaces. At the Centre for Clinical Microbiology, we naturally work very closely with the infectious disease team in the local hospitals. The hospital clinicians who were concerned about this outbreak knew that I've been working on sequencing protocols for various pathogens such as tuberculosis, BCG, which is the vaccine for tuberculosis, and antimicrobial resistant infections in hospitalised patients with COVID-19 in Zambia and Sudan. And they asked if I could help try and identify how the bacteria in this outbreak were spreading. So here is a plain language summary of the research that we did. If you want the link to the original paper, you can find it in the blog. So for a background to the study, antimicrobial resistance is a growing concern globally, and those most likely to be affected are vulnerable populations, such as those living with compromised immune systems, transplant patients, or those receiving cancer treatment. I've written a blog explaining more about how AMR affects people, and you can read it on the link on my blog. Having an infection that doesn't respond to the treatment that it should do makes it much more difficult for healthcare professionals to prescribe the right thing to make you better. Multi-drug resistant organisms, or MDROs, are especially problematic as they have resistances that make them resistant to multiple different drug groups, and this is particularly common in bacteria, and we're discussing bacteria in the rest of this podcast and also in the published paper. They're difficult to treat for the person who is infected with them, but they're also a pain to deal with from the healthcare perspective because they can easily pass their resistance genes between themselves and other organisms, both within the host, the human in this case, and also in non-host environments, such as hospitals, farms, and households. This is why water sanitation and hygiene, or WASH, is so important. In hospitals, identifying multi-drug resistant organisms and their resistance genes in the hospital environment is especially important, as it can help trace outbreaks, and knowing where reservoirs of these organisms are can help healthcare workers target WASH and infection prevention and control procedures into the right places. Many patients who have to stay in hospital are classed as vulnerable. They might be going for surgery or cancer treatment, for instance, and intensive care unit patients are especially at risk. Patients, and people in general, sometimes have multi-drug resistant organisms in their bodies, but they can be asymptomatic and often completely unaware that they have one. Clinicians define this as somebody being colonized by the multi-drug resistant organism, and this is different to being infected. And the infection, or the word infection, means that you have symptoms of that bacteria and are likely to require treatment. If you have an infected patient, so for instance they have a pus-filled wound or diarrhea, then it's relatively easy for clinicians to identify them and take a sample, get a diagnosis, and then treat them. But if a patient is colonized, it's not so obvious. You have to screen all patients, even those who appear healthy, to look for the multi-drug resistant organisms they might be carrying. This can be costly, but colonized patients are capable of transmitting their multi-drug resistant organisms to other patients who might then become infected and therefore sick. The carbapenem-resistant Enterobacteriaceae, or CREs, are a group of bacteria that are causing real concern among scientists and clinicians, and also in the vet world too. Common infectious bacteria such as E. coli, Klebsiella pneumoniae, Citrobacter frundi, and Enterobacter cloacae, amongst others, can harbour resistance genes that make enzymes called carbapenemases. These carbapenemase enzymes inactivate carbapenems, the drugs, and other beta-lactam antibiotics, including penicillins and cephalosporins. This is a really big proportion of the commonly used antibiotics that don't work against these CRE bugs. There are a number of different groups of genes that can cause carbapenem-resistance. These include New Delhi metallo-beta-lactamase, or NDMs, Verona integron-encoded metallo-beta-lactamases, or VIMs, imipenemonases, IMPs, and oxacilinase 48, or OXA48. And these, as you can see from some of them, are often named after the place that they were first discovered. Knowing what these genes are and how these multi-drug resistant organisms can move between people, the environment, and animals is vital to help scientists trace transmission routes and support antimicrobial stewardship. In fact, genomics, the study of genetic material, and sequencing, the method used to identify the order of the genetic material, is becoming more and more important in infectious disease and public health science. One hospital in the UK found that it was having an outbreak of carbapenem-resistant Enterobacteriaceae, both in colonised patients who they had screened and also patients who had become infected and sick with CREs on a ward. They did some rapid testing using something called the NG-Carba5 panel, and this is a bit like the COVID-19 rapid diagnostic test or a pregnancy test. And they found that the patient's multi-drug resistant organisms all tested positive for BLA-OXA48, which is the gene that carries the OX48 oxacilinase enzyme, and some were also positive for BLA-NDM. The aim of our study, the clinicians came to us at the Centre for Clinical Microbiology to see if we could use sequencing to try and track these CRE organisms and their genes around the ward. They wanted to know if we could identify whether there was transmission of these organisms between the patients, but also between the patients and the ward environment. Methods. We collected 28 bacterial isolates from all of the colonised and hospital-acquired infection patients who had tested positive for OXA48 using the rapid test. We then plated these samples out on agar plates and grew them up in special selective media, which helps to give us the specific organisms that we were looking for. We then used something called a mass spectrometer, or MALDI-TOF, to identify these species more conclusively. Mass spectrometry can identify species by ionizing samples into charged molecules and measuring the ratio of their mass to charge. Each species gives a slightly different reading, and the MALDI-TOF is appropriate to analyze biomolecules like peptides, lipids, polysaccharides, or other organic macromolecules. We found, using the MALDI-TOF, that most of the clinical isolates seem to be E. coli, Klebsiella pneumoniae, Citrobacter frundi, and Enterobacter cloacae. We then extracted the DNA from the bacterial cells by breaking the cell walls down using enzymes, and we did whole genome sequencing on each sample using Oxford Nanopore Technologies sequencing platforms. This meant that we had multiple copies of each base in the entire genome, and most enterobacterial genomes are about 5 million bases long, and we could then make sure that we were confident that each base was correct at each point in the genome. We also collected 216 environmental swabs and wastewater samples from the ward where the outbreak was happening. We swabbed lots of different surfaces, including telephones, toilet bowls, bathroom floors, and collected wastewater samples from sink U-bends all around the ward. We isolated 47 bacterial species that matched the clinical species, and these environmental samples were then processed in the same way by performing whole genome sequencing. What we also did is extract DNA directly from the swab and wastewater samples, and this is so we could do metagenomic sequencing. This method is where you take all of the DNA in a sample and amplify it using PCR so that you have enough DNA, and then sequence everything. In theory, you will sequence all of the different species that was found in, for instance, your plug hole, in the same ratio that they were in real life, so living in the actual plug hole. We did metagenomic sequencing for 36 environmental, wastewater, and swab samples, and these were the ones that we had identified and isolated CRE bacteria from. We then had lots of sequencing data to process and analyze. For the clinical environmental isolates, those single species, we looked at the antimicrobial resistance genes that were present and created a phylogenetic tree to look at relatedness so that we could see which isolates were more similar to others and try and understand the transmission patterns. Results. In total, we had good enough sequencing data from 25 of the clinical isolates, and we found that 84% of these clinical isolates had at least one BLA-OXA gene. Now, there are multiple slightly different versions of which OXA48 is the most common, and 76% of the clinical isolates had at least one BLA-NDM gene, and again, similar concept, except NDM1 is the most common one. When we looked at the environmental data, we found that CREs were most commonly isolated from environmental wastewater samples, so in 52%, and then from stick swabs, so those are the ones like cotton buds, from 11% of those, and then sponge swabs, and that was 4%. 18% of the environmental isolates that we collected had at least one BLA-OXA gene, and 2% had a BLA-NDM gene. We also identified BLA-OXA genes in 5% of the metagenomic environmental samples, where the DNA was extracted directly from the wastewater or swab. When we built phylogenetic trees for each of the species, we saw evidence for both patient-to-patient transmission and patient-to-ward, or possibly ward-to-patient transmission too, although this appeared to be less common. Discussion. From the results of our study, we concluded that CREs were more commonly isolated from water samples than either stick swabs or sponge squabs, which means that in the future, when swabbing wards to look for multi-drug resistant organisms and resistance genes, healthcare workers might want to focus on wastewater. Whilst all patients in this study tested positive for OXA48 like carbapenemase variants using the rapid Carba5 test, not all of them, 84% of these patients, had a BLA-OXA48 gene identified in their genomic data. This difference may be because the bacteria are able to take up and expel these resistance genes relatively easily, and in fact they're costly for the organisms to produce, so if they aren't needed they can be spat out. There was a similar story for these clinical isolates regarding the BLA-NDM genes too. BLA-OXA and BLA-NDM genes were less commonly identified in the environmental CRE isolates compared to the clinical isolates, which suggests that there are different populations circulating in patients and the environment, or that there's a greater loss of AMR plasmid genes due to less antibiotic selective pressure in the environment, or possibly that there's a gain of these genes in isolates that live in patients who are likely taking antibiotics, and therefore there's a pressure. We didn't find as many resistance genes in our metagenomic environmental samples, and so we concluded that it did not appear to be as sensitive as isolating specific species in this study. In the future, we can try and optimize this, so to see if we can pick up more, and this might mean taking larger samples or filtering the water, for example, to make sure we're picking everything up. In conclusion, we found that using Oxford nanopore sequencing to identify multi-drug resistant organisms and resistance genes can speed up clinical decision-making whilst awaiting reference laboratory results, which can provide both economic and patient care benefits. As a result of this project, we're planning to expand our genomic search for AMR genes and into other environments, such as veterinary centres. This work has also resulted in me writing up a fellowship application to further develop this sequencing tool for use in low and middle income countries.